Yep,
Wonders.
I got it. Thanks.
Great. Well, thank you all for coming today. I'm Katie Wagner. I'm a member of the Society for the Study of Evolution's Council, and that's how I'm here today to introduce the seminar speaker and the seminar series. Um, I'm here to welcome you to the fall semester series of talks given by SSE's Graduate Research Excellent Grant awardees. One quick note is that the seminar series follows the SSE Code of Conduct, and Robert is going to put that Code of Conduct in a link in the chat so you can review it if you'd like to. This semester's speakers for the seminar series are winners of the 2022 and 2023 Rosemary Grant Advanced Award. And this is the first seminar series we've given for Rosemary Grant awardees, so this should be really fun and exciting. So these awards are for students in their final years of graduate work. The series is organized by SSE's Graduate Student Advisory Committee, also known as GSAC. And a special shout out to Robert Driver for organizing and spearheading this series. And many thanks to all of the hard work put into the series by GSAC that spotlights this outstanding graduate research in our field. It's a really fantastic way to highlight this work being done by graduate students. So today's seminar will be by Megan Molinari from the University of Santa Cruz. Megan is a PhD candidate in Dr. Suzanne Alonzo's laboratory, and she's interested in how highly conserved hormonal and neural mechanisms can contribute to the large diversity of social behavior observed in animal species. Megan did dual undergraduate degrees at the University of New Hampshire in marine, estuarine, and freshwater biology, and in neuroscience and behavior. And that seems like a perfect complement of background to head into what has become her PhD work. She moved straight from her undergraduate degrees to the University of Santa Cruz to begin her PhD work, where she's done extensive field work based in the Mediterranean, based from a field station in Corsica. And hopefully we'll get to see photos of this, what I imagine to be a spectacular place and some really beautiful fishes in today's talk. So Megan will be talking today about her research focused on how hormone manipulations impact the social behavior and fitness of a wrasse species with complex social behavior. She'll then discuss comparative research on neural gene expression of hormone receptors in 5 wrasse species. Her goal is to understand the role hormone expression plays in the evolution of complex social behavior. And with that, I will hand it over to Megan. Her talk today is titled The Evolution of the Hormonal Mechanisms Underlying Complex Social Behavior in a Group of Mediterranean Wrasses. Megan, thanks so much for being with us today.
Hi, thanks so much for that great introduction, and thank you all for being here today. I'm going to share my screen. All right, can everyone see just the slides and not my presenter notes? Great. All right, yeah, so I am giving this talk on my dissertation research on the evolution of the hormonal mechanisms underlying complex social behavior. An overview of this talk. I'm going to start out with a general introduction and introduce my overarching research question. Then I will go over the first 2 chapters of my dissertation. So the overlying focus of my dissertation is social behavior and the mechanisms that control it. So social behavior is really complex across animals. We see everything from individuals who spend most of their lives highly solitary to those who live in very complex social groupings. And throughout animals, what we find is that social behavior plays a really important role in fitness. And so it can impact an animal's reproduction, it can impact competitive interactions, and it can impact cooperation. And so it plays this really important role in fitness. Similar to other traits, social behavior evolves due to the mechanisms that control it, so mainly due to the selection on the neural and endocrine systems. So you'll see things like changes in brain organization or the sizes of certain brain regions with differences in social behavior. And then you'll also often see changes in the hormone systems. And this often takes place as changes in either the amount of hormones that we see or the amount of hormone receptors we see and where they're located in the brain. I'm going to focus in this talk about the evolution of these endocrine systems, because hormones play this really important role in social behavior by allowing an animal to respond rapidly to a changing social environment. Here are some hormones that play an important role in social behavior. They are not exhaustive by any means, but they are the ones that I'll focus on in my dissertation. And so two of the major ones are oxytocin and vasopressin, and these are very closely related hormones that play an important role in a lot of complex social behaviors. So social bonding, a lot of reproductive behaviors, things like that. And then we have sex hormones, which universally have a pretty strong effect on an animal's social behavior. And overall, these hormones together and alone can facilitate a lot of behaviors like social recognition, aggression, and cooperation. And what's so remarkable about these hormones is that they are so highly conserved across animal species. And so what this chart is showing here is it's showing the evolution of these oxytocin and vasopressin system in animals. And so you can see that for the bottom half of this tree here, talking about vasopressin or vasopressin, there starts out with this ancestral vasopressin all the way back in lamprey, and even before this, it's just where this tree starts. And then you see remarkably similar copies all the way up to chickens and other bird species. And then in mammals, we get this version of vasopressin called vasopressin. And a lot of these slightly different versions arise through gene duplications or very small differences in the amino acid sequence. So they're really, really remarkably conserved. And you can see the same thing with the oxytocin grouping. So we see isotocin in our bony fishes, mesotocin in a lot of birds and reptiles, and you get to oxytocin in our mammals. And not only are the hormones that facilitate social behavior really highly conserved, the brain regions that respond to these hormones are also remarkably conserved. And so what we see is that there are 6 major brain regions that form the so-called social behavior network in mammals. So these brain regions light up in response to a number of social behaviors. So aggression, sexual behavior, pair bonding, these same brain regions are involved in all of those behaviors. And what's really interesting is that when you look across animals, we find similar versions or exact copies of these brain regions in other species. And so on the left here, it's showing the brain regions that are involved in producing a type of social call. And so when you stimulate these brain regions with electricity, it causes the individual to produce what's called a fictive grunt. And so these grunts are used in social behavior, particularly in territory guarding. And so these same brain regions, or homologs of these brain regions in fish, are used in social behavior all the way from fish up to mammals, where we see this social behavior network. So my research really focuses on, given how conserved these neural and hormonal mechanisms behind social behavior are, how do they contribute to this really wide diversity of social behavior we observe in animals? Now I will outline some of the research that we know about how these hormonal and neural mechanisms change in individuals with different social systems and social behaviors. So some of the most studied groupings when it comes to social behavior and hormonal mechanisms are rodents, and particularly microtein rodents, which include these 2 species right here. We have prairie voles, who are socially monogamous, and so they form these really strong pair bonds with mates, where you see that characteristically they prefer to spend time with their bonded mate than they do with strangers. On the other hand, we have montane voles, who are pretty closely related to prairie voles, but they are polygynous and they lack these pair bonds. So they do not have this formation of a pair bond where they prefer to spend time with a certain individual over a stranger. And what you see in these 2 species is that there are differences in their oxytocin receptor location and density in the brain. that may facilitate these differences in social behavior. And so when you stain this brain looking for oxytocin receptors, so receptors that bind to and respond to the hormone oxytocin, the prairie voles have the highest amount of bonding in the prelimbic cortex, the nucleus accumbens, and certain areas of the thalamus and the amygdala. And you'll notice that some of these areas are ones that I've stated earlier as being really important in this social behavior network, and some of them are just related to this social behavior network. The montane vole, on the other hand, shows very different patterns of oxytocin receptor expression. So you see a lot of binding in the lateral septum, in the hypothalamus, in a completely different region of the amygdala. So there's very little overlap in the most common binding sites in these two species. So researchers thought that this might play a role in the large differences in social behavior between these two species. And they further studied this by looking at how injections of oxytocin influence these voles' social behavior. And so when you look at the prairie voles, these ones that form this strong pair bond, when you inject an oxytocin antagonist, which will decrease activity, so an oxytocin antagonist will bind to receptors in the brain and decrease the activity of them. And so when you inject this oxytocin antagonist into the nucleus accumbens, this brain area that shows a higher binding in prairie voles compared to meadow voles, you'll find that it prevents the formation of this partner preference. And so the control group is on the left here, and you'll see that normally these voles prefer to spend time with their partner over a stranger. But when this antagonist is injected into the nucleus accumbens, you see the opposite result. So they are no longer forming this strong partner preference. And so this further confirms that the oxytocin system plays a really important role in social behavior and may facilitate the differences in social behavior we see between these two species. So going on to compare this to other rodent species, the picture is not quite as clear. And so when you look at similarly related species and ones with a similar difference in social system, we find that there is this higher expression in the lateral septum for polygamous meadow voles, similar to what we saw in our pine voles. But there's no difference in the nucleus accumbens. So this really distinctive difference in the nucleus accumbens that we saw in the previous study was not found between this monogamous and polygamous species in this case. And there's similar results found in a solitary versus a social tuco, which is another type of rodent. There was no binding in the nucleus accumbens at all for the social species, and there was actually a higher amount of oxytocin receptor expression overall in the solitary tuco. So overall, these studies show that even though the picture seems clear when you're comparing two species, when you start to zoom out, it doesn't seem like there's a very repeated pattern for this hormonal expression and a repeated pattern of where it's located in the brain. And so this begs the question, how repeated is the evolution of hormonal mechanisms underlying social behavior? And do we see the same hormone expression pattern in species with similar social systems? And I think this question is really important because often we use studies on model organisms to determine how a hormone may work or how a hormone receptor in a certain brain region may work in humans. And we're not even really sure whether knowledge gained in one taxa can be extended to others based on how repeated this evolution of these mechanisms are. So I will use this genus of fish species to help dive a bit deeper into these questions. And fish in general are a really great organism to use for studies like this because they have super diverse social systems, one of the most diverse of any grouping, all the way from fish who spend most of their life solitary to those who school and up to thousands of individuals. And fish in general are increasingly used in studies of hormones, neuroscience, and social behavior. This genus in particular is very valuable because it is an adaptive radiation. So all of these 11 species that you see here evolved within the last 1 to 12 million years, and they have a very similar ecological niche. So they have similar diets and they live in similar areas. Many of them are sympatrically located. And what you see throughout that, even though their diets and often their morphology is pretty similar, they have really diverse social and reproductive behaviors. And this takes a form of alternative reproductive tactics, which means that the males in these species have distinct behaviors that they use to achieve reproduction. And this is based on age and size, really. So throughout a lifetime, an individual may transition between these life cycles. And so here are the three alternative types that we see in the system. Not every species has every type. At the very least, they all have this first type right here, which is the territorial males. And so these are the largest and the most colorful male in every species. And they take this paternal dominant role. So they will build a nest where a female can lay eggs. They will guard this nest from sneakers and egg predators. They will court females, so they'll guide her gently towards the nest for external fertilization. And they will compete with other males to fertilize these eggs. Some of these species additionally have this type called the sneaker here on the bottom. These are the smallest type of males, and they often lack color or even look sometimes similar to the females, at least to our eyes. And they attempt to sneak mate into the territorial male's nest. So they have this purely parasitic strategy where they're just trying to sneak in, fertilize the female's eggs, and leave the nest. And then there's this really interesting third type in a few of these species, and these are called the satellite males. They're intermediate in size and color, and they form this temporary relationship with the territorial male that involves both cooperation and competition. So satellite males will cooperate by chasing away sneakers and by courting females, so they will guide a female into their nest. But they also cheat by sneaking into the nesting male's nest and fertilizing eggs when given the opportunity. So this sets up a really complex social dynamic in some of these species. And here's an example of what this looks like in the species Symphotis ocellatus. We have territorial males, satellites, and sneakers in this species. So throughout the Symphotis genus, we see a range of the number of alternative types, all the way from just territorial males in these species marked with an orange triangle. And then we have ones with two alternative reproductive types. We have territorial males and sneakers in these systems, and the ones marked with stars. And then finally, we have two species with this most complex type of social system. So the one with three alternative types that I highlighted. And what's interesting about this tree is that the social systems are not more similar in species that are more closely related. So what it seems like is that the complex social system that we see with three alternative types evolved twice separately in this tree, once in Symphotis ocellatus and once in Symphotis rosali. And this sets up the differences in what challenges the territorial male face. So territorial males in both 1 and 2 type systems really only have to guard their nest and court females. They don't have any additional demands in social behavior. Meanwhile, these two species with 3 alternative types have this additional relationship with the satellite male. And this is a really complex relationship. So the territorial male has to balance the amount of chasing behavior and the amount of tolerance he directs towards his satellite male, allowing him close enough to the nest where he can help guard it, but not too close to the nest where he's cheating all the time and stealing all of the territorial male's reproduction. And so given this complex system where we see the repeated evolution of this type of social behavior, I would like to ask, how repeated is the evolution of patterns of hormonal receptor distribution and expression in the Symphotis genus? And that's what my chapter 1 will focus on. So I kept the prediction for this chapter kind of vague, and that's because I don't necessarily have specific expectations for different brain regions, but I do have a general pattern that I expect to find. And so on the right, or on the left axis, the y-axis right here, you'll see relative mRNA expression, and on the x-axis we have species. And so what I expect is that in a particular brain region, so let's say we're looking at, for example, the preoptic area, looking at a specific hormone receptor, let's say, for example, isotocin. If we're looking at the expression of an isotocin receptor in this brain area, we might find that it is more similar levels of expression between species with 3 alternative types compared to those with 2 alternative types. Alternatively, we might see a pattern where it just is based on how closely related individuals are, or it might depend on the specific brain region and the specific hormone we're looking at. And you'll notice that there's only 5 species on the bottom here, and that's because these are the 5 that are feasible to study based on their densities, where they're located, and other factors like that. So S. mediterraneus, ocellatus, Hinca, and Rosali are all found in Corsica, And then S. melops is found in Norway, and we have partners with people who can send us some samples of them. And for each species, I'm focusing on just the territorial males. Again, because these territorial males are the ones that are really facing these complex social dynamics based on their system. So those in two-type systems have a comparatively simpler social demands compared to those in these three-type systems. And what's great about this spread of species is that I think that there are species from both this top branch, so closely related to the Symphodus ocellatus, and then those kind of on this more bottom branch, Symphodus rosali. So all of this sample collection took place in Corsica, France at the Staressa Research Center. I started out by diving there and observing the behavior of these species. And so I would find a territorial male in the wild, observe its behavior, and watch for some type of spawning behavior. So I'd wait for him to either court a female or actively spawn with a female in a nest. And the reason why I'm looking for a very specific behavioral context is because that can really affect the mRNA levels. So I want to have a similar behavioral context between these species. So after observing this spawning behavior, I catch the territorial male in catch nets. And this often takes, it takes at least two people, sometimes more, to chase these guys down and get them in a net. But within 30 minutes, we have captured these fish, we have transported them back to the lab, and we have dissected their brains. And so you can see a picture of us dissecting the brains. And we just preserve them immediately in RNA later and store them in the freezer because doing brain microdissections in the field is not really feasible where I am. So in the future, what I'm going to do with these brains is I'm going to further microdissect them down into the following regions. And so using coronal sections with a cryostat, I'm going to slice it down really thin. And I'm going to look for these four regions: the dorsal telencephalon, the ventral telencephalon, the preoptic area, and the hypothalamus. So some of these areas are direct parts of the social behavior network, like the preoptic area, and some of them just contain parts of the social behavior network. For example, the dorsal telencephalon contains parts of the amygdala, and the ventral telencephalon contains the nucleus accumbens. And the reason why I'm taking these broader regions is because it's really difficult to get such a fine slicing in brains this small. But getting these larger slices will at least contain these areas of interest. And then I'm going to look at the following hormone receptors. We have our androgen receptors, so those that respond to mostly 11-ketotestosterone is the hormone that's most commonly produced in fish. And then we have our isotocin receptors and our vasotocin receptors, which you can see the sequence of on the bottom left and the bottom right, respectively. And you'll notice that there are often multiple copies of these receptors labeled here, and that's because there's a lot of gene duplication events that occurred in teleost fish, leading to two different distinct copies of these receptors. And they often are involved in different social behaviors. So it's really important to look at these separately because sometimes they can play a different role in the social behaviors. So what I expect to find for this chapter is that if I find similar patterns of receptor expression in species with similar number of ARTs, that means that the evolution of complex social behavior is relatively conserved. So we'll see these same patterns. And further, if these patterns are similar to what is found in other species, for example, if we find that the same brain regions in these teleost fish that are responsible for these complex bonds are maybe found in voles or some other groupings, then this suggests that the evolution of complex social behavior is really constrained. So there's only really a couple of ways you can achieve this complex social behavior. On the other hand, if we see different patterns of receptor expression, even in these very closely related species, and this suggests that there are many pathways to complex social behavior. And what we learn in one species or one genus might not necessarily be extendable to other groupings. And so I've collected all of the samples for this, and I'm currently working on getting training in this brain microdissection. So I'm super excited to get that brain microdissection done so I can send out these samples and see these differences in gene expression between these species. So far I've gone over my broader introduction of this project. I've gone over chapter 1, and I will continue on to my second chapter. So a reminder that my research question is really looking at the evolution of hormonal mechanisms underlying social behavior and whether these patterns are repeatable. And so in chapter 1, I hope to gain insight into how hormone receptors have evolved in a genus with variable social behavior. For chapter 2, I hope to dive further into how these hormones actually impact social behavior. So within this genus, how do these hormones influence social behavior and fitness? I'm going to focus on a specific hormone in a specific species, looking at how the hormone oxytocin influences the social behavior of territorial males in the species Symphotis ocellatus. And this is going to be a really good complement to my first chapter, because it's going to tell me how these hormones actually influence social behavior. So let's say if I find, for example, that oxytocin receptor expression is higher in certain regions in species with 3 alternative types. Knowing how this hormone impacts social behavior might help explain why we see those differences. So why did I choose the hormone oxytocin? This is homologous to the hormone oxytocin in humans, which you may have heard of as the love hormones, because it's really involved in pair bonds and complex social behavior. I also chose it because it plays this really important role in interspecific differences in social behavior, like we saw between our 2 vole species in the beginning of this. And it plays a very important role in social behavior because it's involved in social recognition, the formation of social bonds. And for a long time, it was generally thought to be this really pro-social hormone. So this hormone that increased affiliative social behaviors, increased things like pair bonds. But in recent years, we've become more appreciative of the fact that oxytocin can actually increase aggressive behaviors and kind of have the exact opposite effect on social behavior. And so some new theories on the role of oxy or oxytocin talk about how it might modulate the attention of an individual or control how much attention it's paying towards social cues. And so we don't really know quite how it works, even in humans, but there's lots of possibilities. And why did I choose the species Symphotis ocellatus? This is because it's one of the species with this complex social system with 3 alternative reproductive tactics. And this is really important because what we have in these species is repeated social interactions between 2 individuals. And so territorial males and satellites in Symphotis ocellatus form this temporary relationship that can last anywhere from a few hours to a few days. And so I know that when I capture the territorial male and inject him with a specific hormone, or when I inject him with oxytocin, that when I return him to his nest, he will go back and the satellite male will still be there. And so I'll be able to see their interactions before the injection and then compare that to their interactions after the injection. This is great because everyone else at the nest environment tends to be pretty transient. So females seem to come in and out, as well as sneakers, making it a bit hard to control for individual differences in behavior. But this territorial male in the satellite remains pretty constant. And so I can look at how these repeated competitive and cooperative interactions between the territorial male and satellite change with injections of oxytocin. I start out by finding a nest with both the territorial male and the satellite present. And then I wait to see that courtship or spawning is occurring. So at least seeing a female led into a nest or spawning occurring to make sure that the nest is active and it's in the spawning phase. So spawning is actively going on. And then I make sure that there are at least 2 sneakers present at or around the nest, within 1 meter. And that's so that the activity at the nest is high enough so that when I catch this territorial male and release him back, the activity is likely to rebound and he's not likely to abandon the nest. So when the activity is very low, we'll sometimes see territorial males abandon the nest, which would ruin the experiment. So this also takes place in Corsica, France at the Stressa Research Center, similar to previous study. And once I find these nests, I drop 2 GoPro cameras, one with a really close view of the nest to capture things like spawning, a lot of the territorial male behaviors, and things like that. And then one with a wider view of the nest to capture nest activity. And I let these cameras film the nest for 10 minutes while we swim away and let them remain undisturbed. And this is to look at these individual differences in territorial male and satellite behavior. So looking at kind of these distinct behaviors that each individual has. Once these 10 minutes have run out, I use the same catch nets and methods in the last chapter to catch the territorial male. We then transport him really rapidly back to the lab where we pass him off to my lab mate, Lou, who is a saint, pulls them up, runs down into this lab you see over on the corner here, and then measures them, weighs them, gives them an individual mark so that we can distinguish between them. So we use elastomer, which is like a small dye, to distinguish between the different territorial males. And then she injects them with one of 3 treatments. So it's either isotocin dissolved in saline, an isotocin antagonist dissolved in saline. So the first one should increase isotocin in the brain. Second one should decrease it, have the exact opposite effect. And then finally, control saline. So injections of this control saline is a great control because it shouldn't affect isotocin at all, but it's a really good replicate for this very disturbing or this very disruptive behavior of pulling them away from their nest, having them ejected, and then releasing them back. So then Lou passes the fish back to us for the same method. We swim him really rapidly back to the nest, and always within 30 minutes of capture, we release him back to the nest, as you can see me doing right here. After that, we really leave the nest alone. We drop 2 more GoPro cameras with fresh batteries, and we let them run out. And so what we'll take from that is 2 more 10-minute video clips, 40 and 75 minutes post-injection. And the reason why there's 2 time points here is because we're not exactly sure when the peak of isotocin activity is, but it's likely to fall somewhere within that window. And here are the behaviors I looked at. So taking, after I left the field season, I had 30 minutes of video for each treatment. So 10 minutes of this precondition, and then the 40 and 75 minutes for each trial. And these are the behaviors I analyzed. So I have students that are blind to the treatment analyze these videos to prevent any bias. And at the end, we compare the sum of these behaviors between pre and post conditions between the different treatments. So you sum up the total behaviors in the 10-minute video. And so my main response variables are these territorial male behaviors that I expect to be directly impacted by these injections. So we have chases. So a territorial male will chase away sneakers, satellites, and other species. And this is an overall measure of his aggression. So I'll have overall total number of chases, and then I'll also split that by chases of just a satellite and chases of just sneakers. I also have count the territorial male follows, so the amount of times they follow a female. And this is how they court females. They'll follow them gently toward the nest. We also have spawns, and this is a sort of indirect measure of fitness in this territorial male, because we're not actually counting the number of eggs he fertilized, but we are counting the number of times he reproduced in the nest throughout the 10-minute video. And then I measure a lot of covariates because the behavior of others at the nest can have a large impact on the territorial male behavior. So this includes things like the amount of times the satellite male chased others, the amount of times the satellite male followed females, and the amount of times the satellite male spawned and sneaked, as well as a measure of nest activity. So this is the sneaker and female present at the nest, which really controls how many opportunities for reproduction, how many opportunities for aggression the territorial male has. And so there's a couple of possible hypotheses based on how isotocin acts in these fish. And so if it has this general pro-social effect we see in some species, then I expect that territorial males injected with isotocin will show a greater decrease in the number of chases from pre to post condition compared to territorial males injected with control saline. And so basically they'll show this general decrease in their aggression towards everyone. And so their total number of chases. And you won't see it specific to a satellite or a sneaker, just total chases in general. It's difficult to predict how this might change the number of spawns the territorial male has, so I won't make a specific prediction about that. If instead isotocin has this role as a modulator of social attention, which has some increasing evidence in recent years, then I expect that the degree to which a territorial male chases changes from pre to post will be more closely correlated with the social environment. So for example, I can look at the number of chases compared to the behaviors of the satellite male and compare how they've changed from pre to post conditions with territorial males injected with isotocin compared to those injected with saline. And I further expect that this will probably have an impact on the territorial male's fitness and have a greater increase in response from pre to post condition when injected with isotocin compared to saline. And for both of these, I expect the antagonist to just have the reverse effect of isotocin, as it's decreasing isotocin in the blood. There are other possibilities. For example, in some cases, it seems that isotocin or oxytocin can change behaviors directed towards specific individuals and not others. So, for example, it might control how the territorial male behaves towards males, but not females. But we'll talk about a bit of those later. So far, I have collected what I hope to be enough samples for this. I have 11 trials of isotocin injections, 9 where individuals were injected with the isotocin antagonist, and 12 controlled trials. Just to mention, these territorial males were only used once, so they were injected with this, and since they have this individual mark, we chose fully different males for each trial. And this was collected over 2 years because these trials take quite a bit of time. And my students have completed over 3/4 of this video analysis. It's a lot of footage and it takes a long time to do. And so the only thing they have left to do is take a really deep dive into these wider views that I mentioned and looked at these nest activity variables. And so all of the results that I'm going to be presenting are very preliminary because nest activity is super important in behavior. And so while I can kind of explore the data and look at some preliminary relationships, they are warranted to change based on these nest activity variables. And so here are some graphs, and these are showing the different scores. So they're showing the change in these behaviors from pre to post conditions. And we have both post 1, which is 40 minutes after injections, and post 2, which is 75 minutes post-injection. And the reason why we have both of these again is because we're not sure when the peak of activity of both the isotocin and the antagonist are. So in this most left graph here, we see the change in territorial male chases, so his total number of chases from pre to post, and then the different treatments on the x-axis. And so what I found with a preliminary analysis is that the number of total territorial male chases were not significantly affected by treatment. Kind of matches what you see on the graph. And you see the same pattern for territorial males following females. There's also a similar trend that I saw with change in territorial male spawns. But I found 2 borderline significant effects as I was exploring this data. And the first was in the number of time a territorial male chases his satellite. And so not looking at total chases, looking at how much this territorial male chases the satellite in particular. And this shows a greater decrease from pre to 75 minutes post-injection in antagonist trials compared to control. And so in this case, it seems like territorial or the isotocin antagonist lead to a further decrease in territorial male chases. And when I further looked and tried to look at the relationship between territorial male chases and satellite behavior, I found that the number of territorial male chases increases more in response to satellite behavior, 75 minutes post-injection, the isotocin, compared to control saline. And so what we see is an increase in the number of territorial male chases compared to satellite behavior, 75 minutes post the injection of the isotocin. But both of these are very weak effects, and I expect them to be more clear once we have these measures of nest activity. Yeah, and so looking at these sort of weak effects, the reason why this, I expect that we're seeing such weak effects is that a lot of these variables are highly correlated. So this is super common in behavior, that all of these behaviors are linked biologically. Yeah, and it makes sense that we see strong correlation, for example, between the number of sneaker sneaks and the number of satellite sneaking, because those are based on both directly on how the presence of females are used. And so running this, I ran a preliminary PCA. What I hope to find is that once I add these variables of nest activity, so the number of females around and the average number of sneakers around, it will help make these PCAs even stronger. It will reduce some of the variation in the dimensionality in the data set. And it's likely to make some of these relationships that I'm seeing more clear. But to take some takeaways from the effects that we did find, I found little support for a general prosocial effect of isotocin. There was no effective treatment on the total number of territorial male chases. But there was some weak support for isotocin as a modulator of social attention. And this is because we found that the treatment seemed to affect very specific measures of territorial male aggression, so specifically his aggression towards the sat, where an antagonist decreased the number of chases that we saw towards the satellite, and isotocin actually increased the number of territorial male chases relative to sat behavior. So it may have matched the number of territorial male chases to the satellite behaviors. And there was no effect. But one thing that I was expecting for this hypothesis, for the idea of isotocin as a modulator of social attention, is that there would be some impact on fitness. And we found no effect of these treatments on total response. And so it's not super clear that this is true one way or the other. Another possibility is that isotocin may just play a larger role in modulating interactions in an established relationship. So it may play a large role in this relationship between the satellite territorial male and not play a role in these relationships of the territorial male with relative strangers. So both the sneakers and females come in, they come out. It's a very transient relationship compared to this established relationship between the territorial male and the satellite, which involves repeated interactions. So overall, sort of unclear, but definitely some possibilities for some interesting conclusions here. And so to summarize, in chapter 1, I will look at how repeated is the evolution of hormonal mechanisms underlying social behavior. And in chapter 2, I'm going to dive further and look at how hormones influence an individual's social behavior and fitness in the wild. Some takeaways from what I've learned is that the impact of hormones on social behavior is complex and can vary widely based on the type of social interaction occurring. And so wild-based studies like the ones I did are super valuable because they allow us to study individuals over a wide range of social contexts. So for example, if you're studying an individual in the lab, you might just be able to see his interaction towards the female or towards a very limited number of social interactions. But when you're studying them in the wild, you can see the full range of natural social contexts they're in and see how this hormone is impacting their behavior in these cases. And I hope to one day extend the study of isotocin on the social behavior of Symphytis osculatus to other in those genus. So see if others in the Symphytis genus show similar impacts of behavior from isotocin injections. So the significance of this dissertation, my first chapter really looked at, or my second chapter really looks at how the behavioral and fitness effects of a hormone in one species. And this will help us develop an overarching theory on how that hormone influences social behavior in other species. So do we see similar patterns in how a hormone impacts social behavior in one species, or does it seem to be very dependent on the species? And then further, I hope in chapter 1 to understand how patterns of hormone receptor expression in the brain have evolved. And this will help us understand how constrained or flexible the evolution of social behavior is, and whether we see predictable patterns in species with similar social systems. And with that, I would like to thank my advisor, Suzanne Alonzo, my lab mates, past and present, Sabrina, Regina, Matt, Lou, and Maddie, and then people who have helped me out in the field, in this picture on the bottom here. And then a huge thanks to the funding sources that made this work possible: the National Science Foundation, University of California, Santa Cruz, the Animal Behavior Society, and of course, a huge thanks to the Society for the Study of Evolution for making the mRNA comparison I'm doing in chapter 1 possible. And with that, I will take some questions.
Thank you, Megan, for a wonderful talk. That was great. So we'll open it up to questions. You're welcome to put questions in the chat and I can read them for Megan, or feel free to unmute yourself and ask away. I can get us started just to warm up the room. So these isotocin experiments are really fascinating, and it's really cool to see those initial results. I'm just curious if you could tell us a little bit about how the fish respond to these treatments. Does it take them a long time to like get back to their normal wrasse lives once you put them back in the water, or what are your observations there?
Yeah, that's a great question. And that is why having this control group is so important. So let me try to go back some slides and take a closer look at these results slides. So it definitely takes them a while. So sometimes we release them and they actually go right back to the nest, and we're like, whoa, like you figured it out. Sometimes they'll swim off into the seagrass, and usually we watch from kind of further away just to make sure they do return. And sometimes it'll take, I mean, at most, probably 5 minutes for them to return back to their nest. And often they'll just hide in small crevices and things like that, and then return back to their nest. So they all return to their nest pretty rapidly. What gets pretty disruptive is that sometimes, especially towards the end of the season, a new territorial male will come in and try to take over the nest. And so they'll go back to their nest and immediately have to fight off another territorial male trying to take over the nest. And in one case, there was three territorial males at once. And so this poor guy got injected, came back to his nest, and was like having this battle with three territorial males at once. Um, and so they do seem to return to the nest. And after a bit, they do return to what is normal social behavior. So they'll do the spawning, they'll do the following the females, and they'll do the chasing behaviors. But it's usually a lot less. And so I'm still trying to determine whether this is due to a drop in nest activity or whether this is due to, just the effect of the treatment. But you can see that all of these y-axes right here are either close to zero or negative. And so there's a general drop in behavior. Even for these control groups, there's a general drop in like overall amounts of behavior. So yeah, that's why it's good that we're comparing to the control, because they do get pretty impacted by the treatment alone.
That's really interesting. Oh, Amber has a question.
I keep looking for the how you raise your hand thing, and I don't know. I can't find it. It's
I can never find it either.
so Megan, this is so cool. So this project you're doing in the system that has the three different the sneakers, the satellites, and the territorial. So do you have any thoughts about doing it with the in-between one where it's just the territorial and the sneakers, right? Is that right? Or do I have it?
Yeah.
Yeah.
Yes, I definitely have thoughts about doing that. And so I would love to do it in some of these other species that have just territorial males and sneakers. Some of them are just methodologically harder. So for example, the Symphytis ocellatus is the easiest to catch, and so it probably takes us at most 10 minutes to catch them. With the Symphytis tinca, they're really big and so they're fast. And so it takes like at least three people when you're chasing them down. And at that point, they're stressed. And same with Mediterraneus, they're just kind of smart. Like they're really good at escaping. And so I would really be interested in how it might change the behavior in these different systems. But as of now, the most feasible one is Symphytis ocellatus till we figure out how to better catch these other ones.
Interesting.
Yeah.
And can I ask you one more question really quick?
Of course, yeah.
What do the asterisks mean on the phylogeny? I was curious when you were going through.
So some of these, yeah, the asterisks mostly mean that the sources describing their social systems are a bit contradictory. And so most of the ones in this study are ones I've observed directly or can have observed directly. So I'm going to have some video of SMELOPS sent to me just so I can confirm. But sometimes what you'll see is that in certain textbooks, they'll describe the social system in one way, and then in another, they'll describe it a completely different way. So those are ones that I, yeah, have some questions about.
Okay, cool.
Yes. Mhm.
One question kind of related to that is there any evidence for intraspecific variation in the social system? Because it seems like I'm just curious how easy it is to switch between social systems if we have this much variation across the phylogeny.
Yeah, great question. As far as like large differences, for example, in the number of alternative types, it doesn't seem like there's a big difference in that. And so I think honestly, some of the reasons why these social systems evolve in certain species is some of them have sex change and some of them don't. So for example, in Symphytis mediterraneus, in Symphytis rostratus, we really only see this because there'll be females when they're small and then they'll transform into males when they're larger. And so there's really no presence of sneakers. And kind of throughout, we see that there's no like a satellite male won't show up in a species where satellite males haven't been described yet. But as far as differences like within the types, there is a wide variety there. And so sometimes we'll see, for example, a satellite male that will try to like take over a nest and will try to do some of the characteristic territorial male behaviors. It's just he's not very good at it. So there's something about how they developmentally change between these types that satellite male can't really do the building behaviors that we see characteristics of a territorial male. And if he tries to take over a nest, like females aren't very interested in him. They definitely have a strong preference for territorial males. And so he won't be very successful. So yeah, definitely some variation in how these fish are behaving within types, but not a lot of variation in these larger social systems.
Yeah, I think what Katie's asking maybe, or maybe if from my understanding, is if is it possible to have a tip on the phylogeny that has like a triangle and a star?
Oh,
Some places it's triangle, some
hmm.
places it's square in the populations.
Oh, that's a really good question. I guess my best answer would be not that I've heard about. So I guess maybe that is a possible explanation for why I'm seeing um,
In the literature?
different literature sources. Yeah. But as far as I can tell, where these guys have been actually directly observed and characterized very completely, it seems like their behaviors are very consistent, but that's definitely a possibility. And I imagine, especially in places where, I don't know, the population is stressed or not doing very well, you might not see these characteristic social systems that we see here. But I'm not quite sure on that.
And I just wanted to ask when you put the fish back and then there's three males competing, uh say that the male that you originally there goes off to some other nest and is replaced, or it doesn't even matter if you took it away. Say just in nature it's replaced by another male. Do the satellites, are they faithful to the nest, or they go or faithful to the fish, or do they not even really care about either? They'll
Yeah, really good question. I would say the satellites are more faithful to a nest than they are to the fish. So they would never follow a territorial male that got chased away. Because in that case, the territorial male has kind of lost, and he'll often go and like wander around until he builds a new nest. And so there's not really anything for the satellite there. Whether he remains at the actual nest kind of depends on the territorial male. So the new one that took over might accept the satellite male, might allow him close to the nest, or he might chase him fully off.
And I just one more thing. You said that sometimes you're down there with the nets trying to catch the fish for half an hour. It can take half an hour. But some of them maybe you catch like really quickly. Like
Yeah.
it's not that hard. So like, do you think that will affect your results? How long it took?
Yeah, so with as far as the actual study went, all of these fish were probably caught within like 5 to 10 minutes. I don't know if I have an exact timeline, but if they take a really difficult or a really long time to catch and it's really difficult, we just abandon it because at that point they are really stressed and I
I
imagine
see.
it could affect the expression. But when we've been trying to catch these guys in the past just to practice it or do other sort of manipulations with them, yeah, some of the fish are much harder to catch than others.
Gotcha.
One more quick one from me. Can you tell us about the geographic distribution of these species? You mentioned one in Norway and the rest of them in the Mediterranean, so that made me curious where they live.
Yeah, good question. So Symphodus truta and Ceralius, I think that's how you say it, live in the Azores. Symphodus melops is the one found in, I believe it's Norway. It's one of like the Scandinavian countries. And then I think all of the rest of them are found in at least some density in Corsica. So they're all Mediterranean species. And so I know the radiation originally evolved in the Mediterranean Sea. I actually don't know much about how these species ended up in different places or what the pathway went like that. But I definitely should dive a bit more into that.
Great. So out of the Mediterranean radiation is what it sounds like.
Yeah.
Yeah, cool.
I have one more. This is my last one.
Awesome.
Do you think there's a difference between the sneakers that are in a satellite system versus a non-satellite system? Do they have to deal with more when they're having satellites to deal with?
Yeah, I've never really thought deeply about it, but I think that you're very right about that, that a sneaker in a system with these two types have to face more challenges. And you do see some differences in behavior. So in some systems, a sneaker will really just come in and leave and be completely gone. So some of these two type systems, like Timidus tinca, it's highly opportunistic, and then they just go and find another territorial male. But we often see in the ocellated wrasse or Symphytis ocellatus and Symphytis rosali, these three type systems, sneakers will hang around really far away from the nest and do
Hmm,
repeated attempts to get there.
hmm,
So I imagine it's much more difficult to deal with both the satellite pressure and the territorial male, both at the same time.
interesting. Thanks.
Any final questions for Megan in our last minute here? If not, let's thank Megan again for a wonderful talk. And I think I can speak for everyone when I say I'm really excited to see how your results go. So we'll be on the lookout for your talks at evolution meetings in the coming years as you wrap things up. Thanks,
Yeah.
Megan.
Thank you so much for the great questions and being a good audience. Thank you.
Thanks a lot, Megan. Really appreciate you presenting. Thank you, everybody, for attending.